There seems to be a crossing line that all living organisms on the Earth have to cross one day. Death. Nobody can escape the inevitable. We are destined to die. Life is a trajectory from the moment we are born to the moment that we vanish from the face of this Earth. Time moves without any compromise. Aging is as certain as the sunrise and the sunset, the seasons and the moon cycle. We see our grandparents and parents age before our own eyes, and ultimately we see ourselves age. There seems to be a sand clock that runs out eventually. But what if I told you that scientists who study the biology of aging have actually figured out a way to reverse aging altogether? Have you ever heard of the Japanese researcher Shinya Yamanaka? Shinya Yamanaka discovered how to convert adult cells into pluripotent stem cells by introducing 4 specific genes called OCT34, SOX2, KLF4 and c-MYC. These Yamanaka factors allow mature cells such as skin cells to revert to an embryonic-like state, where they can develop into any cell type in the body. And this breakthrough, for which he won the Nobel Prize in Medicine in 2012, revolutionized regenerative medicine by offering a new way to generate stem cells for disease modeling, drug screening and potential cell therapies without the ethical concerns of using embryonic stem cells. Yamanaka's work demonstrated that it's possible to reprogram mature cells to an immature pluripotent state, similar to embryonic stem cells. The ability to generate any cell type from a patient's own cells offers vast potential for regenerative medicine, including repairing damaged tissues and organs, and maybe reverse aging altogether. But there is a big problem with this discovery. The 4 Yamanaka factors are enough to remove the genetic roadblocks that prevent a fully differentiated cell from suddenly changing back into a stem cell. But the process is very time-consuming and has about 1 in 100 chance of success. So, in a nutshell, it is very ineffective and it is very costly to do any research. And hence, scientists try to find ways to improve the Yamanaka factors by mutating the genes. And they successfully improved the effectiveness of the stem cell inducing cocktail, but it was still not enough to make a huge difference. So, for a long time, the research stalled with little advancement, and one of the greatest discoveries of the 21st century vanished from the headlines. Now, fast forward to the year 2025 and the introduction of large language models by OpenAI. They revolutionized our understanding of language and invented on the side an artificial intelligence that changed the fate of humanity forever. Before we dive deeper into the enhancement of the Yamanaka factors, let's examine together how large language models actually work. You see, large language models, or LLMs, are AI systems trained on massive amounts of text data to understand and generate human-like language. They work by predicting the next word in a sequence, but through massive scale. Billions of parameters and huge datasets that form connections between tokens called weights. This allows them to develop abilities like answering questions, writing code, summarizing, reasoning, and more. Their algorithm learns to make the next most probable prediction. It can predict, depending on the context and based on its calculated weights, what the next information will be. Others might argue, bluntly speaking, that LLMs are capable of learning and cognition. Let's summarize this again because it is so important. Large language models, LLMs, are AI systems trained on vast amounts of text to predict the next token in a sequence. Through billions of parameters that encode statistical relationships, they capture enough structure in language to generate fluent text and perform tasks like summarization, reasoning, and coding. While fundamentally based on probabilistic prediction, their scale leads to emergent behaviors that resemble understanding and problem solving. Though whether this counts as genuine cognition is debated. And this is why you can enter a bad written text into ChatGPT and it will write a way better version. Not because it is intelligent, but because it has internalized the best statistical probability of the next word of your text based on the whole literature of the world. This allows the model to even imitate certain author styles. Now let's use our new understanding of large language models and apply it to biology. Proteins are chains of amino acids, 20 letters like a biological alphabet. Just like words in a sentence, the order of those amino acids matter. Instead of predicting the next word, a protein LLM predicts the next amino acid. By studying billions of natural protein sequences, it learns the grammar of biology. The hidden layers of the model capture biochemical properties like structure, folding patterns, and binding sites. This concept is similar to how word embeddings capture the meaning of human language. So in essence, protein LLMs treat amino acid sequences like language, learning the syntax and semantics of life, and then using that knowledge to predict structure, function, or design new biomolecules. Now let's go back to the Yamanaka factors and see how AI has advanced the field of stem cell research. The company involved in this research was OpenAI. The company teamed up with a startup to train a new language model called GPT-4b Micro. It was trained on datasets combining protein sequences, structural annotations, evolutionary relationships among proteins, and functional information. The model was then prompted to generate variants of the Yamanaka factors. It introduced mutations and even redesigned two of the Yamanaka factors, SOX2 and KLF4, which they called RetroSOX and RetroKLF. These variants differ from the wild type, sometimes by many amino acids, and even changing a third of the amino acids in some cases. The approach is somewhat like few-shot prompting to direct the model to propose changes aiming for better function. After generating many variants, these are tested in vitro in cell assays for how well they perform, like how much or how fast they induce pluripotency markers, how robustly, how many cells are reprogrammed, and the speed of marker appearance. They also tested for other properties like DNA damage repair, since aging or cell damage is often tied to DNA damage and genomic stability. What was achieved from the initial reports? The AI-designed variants RetroSOX and RetroKLF outperformed the wild-type versions in many of the lab assays. In fibroblasts, combinations of RetroSOX and RetroKLF led to faster and stronger expression of early and late pluripotency markers compared to the original Yamanaka cocktail. Some markers even appeared earlier. In DNA damage repair assays, cells with the engineered factors had fewer double-strand breaks relative to those reprogrammed with wild-type factors. That suggests potential for more rejuvenation or better restoration of youthful cell states. Overall, there is a claim of up to 50-fold increase in expression of some stem cell reprogramming markers when using the redesigned factors versus the original. Imagine what these results mean for the aging research. If we today are able to produce transcription factors that induce a 50-fold increase in expression of some stem cell reprogramming, we are on the path to extend our lives by many years or cure aging altogether. In some years, it could be possible to design a drug tailored to your own genetic makeup to reduce aging effects in your body. AI would be a tool to allow for personalized medicine. While these are promising results, there are still important open questions and limitations. Let's address some of those open questions. The first one being, are these newly designed proteins safe, or do they cause off-target effects? I mean, faster reprogramming is good, but we need to be sure that the variants do not cause unintended effects, like aberrant gene activation, tumor risk, or loss of cell identity. What are the effects of newly designed proteins across different cell types and different donor cells? Performance might vary depending on the cell type, donor age, and delivery method. Some of those were tested, but broad generalization still needs more evidence. How young do the cells actually become? It's not yet clear whether these engineered factors merely speed up the same reprogramming path, or they allow for reaching better, more youthful, more functionally potent end states. Finally, we also need to understand the mutations and redesigns on a molecular level. Understanding why certain mutations improve performance is still being worked out. Which changes affect binding, cofactor interaction, or chromatin accessibility. Why is this discovery so interesting? It shows that protein LLMs can go beyond just predicting structure or annotating proteins. They can design functional improvements in very challenging proteins. It suggests that biology problems that used to require long trial and error like mutagenesis or directed evolution can be accelerated with smarter computational proposals. For longevity research, being able to reprogram cells more efficiently with fewer side effects opens possibilities for better therapies.